Good morning, good afternoon, good evening. Welcome to the second episode in the Waters Biosimilars online seminar series. Today's topic will be LC-MS workflows for biosimilar comparability assessment and will be presented by Samantha Ippoliti from our science team. Please be aware that this is a four-part series. The QR code on the right can be used with your phone to register for the other episodes if you've not done so. And you will receive an email post this event with links to do so as well. I'd like to introduce our speaker today. Samantha Ippoliti is a principal scientist within the Discovery and Development Organization at Waters that focuses on LC-MS workflows. She did her educational training at Northeastern University, graduating in 2012. Went on to 5 years at Novartis in the developmentability assessment role, primarily doing LC-MS characterization of protein therapeutics. She joined Waters in 2018, and in the last 5 years, she's really focused on the LC-MS workflows for biopharmaceutical protein and peptide characterization and attribute monitoring for those molecules. And she's done so on a wide variety of not only our chromatographic platforms, but with the BioAccord, Xevo, Synapt, and Cyclic platforms for mass spec detection. I'll indicate to you that your microphone and camera are switched off by default. If you wish to pose any questions to Samantha, please use the Q&A button at the bottom of your screen. We will, at the end of her presentation, open up a Q&A session where we will hopefully take all the questions that are asked. If not, we'll follow up individually with any questions that we cannot get to within time. A reminder that this meeting is being recorded, so you'll be able to watch it again if you miss any details or refer your colleagues to it after the event. I'll now turn the agenda over to Samantha for her presentation entitled Comprehensive Robust Workflows for Biosimilar Comparability Assessment. Samantha?
Thank you, Scott. Glad to be here today to discuss our workflows for biosimilar comparability with you, specifically focusing today on a newer QTOF instrument, the Xevo G3. So without further ado, let's get started. There we go. Just a quick overview of what we're going to discuss today. First is just typical LC-MS assays for antibodies, mAbs that are typically used for things like biosimilars. We'll look at some solutions from Waters, including the new Xevo G3 QTOF. And then we'll take a deeper dive into a case study of biosimilar mAb analysis in the following ways. So intact and subunit analysis, the native IEX or ion exchange MS analysis, as well as peptide mapping at the peptide level. So what do we normally look for in a mAb analysis with LC-MS? It's becoming a more routine characterization tool for mAb development and characterization monitoring, all the way from development into monitoring and QC. So we need to know what the primary sequence is for the innovator and the biosimilars that we're going to look at. Biosimilars will have the same exact sequence, but they may have some differences in post-translational modifications, which are going to become important in the case study that you'll see today. We look at things like oxidation, deamidation, isomerization, glycation, as well as the N-glycosylation profile that you'll see. And unprocessed C-terminal lysine is a common one as well. And those will all be looked at with LC-MS analysis that I'll talk about in a few minutes. Before I do, just a quick overview of what the Xevo G3 QTOF has the updates that it has from the previous generation. This is kind of our workhorse for LCMS characterization and monitoring. We have updates from the ion optics in spectrum dynamic range enhancement, really improve the sensitivity of the instrument, as well as a few other things like lower in-source fragmentation that we see, and included a 16,000 M/Z calibration for the mass range as well, which is really important if you're looking at different aggregates of maps. So we also look at kind of the whole workflow. We kind of position a Xevo G3 as, like I said, our workhorse for characterization. It's also put onto our Waters Connect platform for informatics. So this is a compliance-ready software that is on the Xevo G3, as well as our BioAccord system, our benchtop TOF system that's really good for monitoring. So because it's on the same compliance-ready informatics platform, it's very transferable if you want to use, let's say, the Xevo for characterization, and then transfer methods to the BioAccord for monitoring. If you have interest in this, we have an app note out to guide you in this analysis and workflow for monitoring. Okay, so our case study for today. We're going to be looking at Remicade as our innovator, and that's infliximab, and then 3 of its approved biosimilars. So that's Inflectra, Avsola, and Renflexis. We're going to be looking, like I said before, at reverse phase intactin subunit. Then we're going to be looking at a native ion exchange MS technique in intactin subunit, as well as peptide mapping and MAM. So we're going to be characterizing and then looking at targeted monitoring of some of the product and process quality attributes. First off is intactin subunit reverse phase analysis. This is a denaturing technique. First slide I have on this is just showing me the intact MAB. On the left side is the combined raw MS spectrum from Remicade on top in red and Renflexis in blue, which is the biosimilar. One of the biosimilars that's, let's say, the most different. And we have a zoom of one of those charge states. And then on the right side, we have the Maxent1 deconvoluted spectra for each one. You'll notice that I've got little blue stars on there on some of the C-terminal lysine variant peaks. And that is where you're going to see the biggest differences for this innovator and biosimilar. And we'll get into more details on that soon. This mirror plot was generated in Unify analysis, the Unify app analysis. So we'll get into some of the other informatics techniques we have in a few minutes. One of those is IntactMass app. This is a relatively newer app that we have within Waters Connect. It does automated peak picking for you in the UV anti-IC. It actually does automated deconvolution by generating the optimal input and output mass ranges and deconvolution conditions. So it really tailors it to that one injection and that analysis, or your specific data, right? So it's going to do give you the best possible deconvolution that you can get for that injection. It does automated acquire and process. So within the same app, you can acquire your data and process it at the same time, like as it's going. It also has the option to assign some IDs using variable modifications. If you're familiar with Unify or other software products that you put in modifications and try to mass match it, that will do that for you as well. It has the capability of using BaySpray or Maxent1. This is the table on the right side shows you the details of what it's going to be doing. So for intact MAB, we're going to be usually using Maxent1 for average mass, and that's in the range of 15 to 300 kilodalton proteins. Okay, here's just an example of what you get in an untargeted analysis with intact mass for our 4 biosimilars. Remicade on the top left, followed by Inflectra on the bottom, Avsola on the top right, and Renflexis on the bottom right. And you can already sort of start picking out some of those differences in the C-terminal lysine variants. And you can see a nice glycoprofile, as you look and compare the differences. Okay, so from here, we wanted to look at an intact MAB glycoprofile. So in order to do that and make it simpler, we did a carboxypeptidase B enzyme treatment that removes all of that unprocessed C-terminal lysine that makes it more complicated. So we could really compare the profiles at an intact level. And so what you see here is the plotted N-glycan forms. And this was a triplicate injection, so the error bars there represent that the error in the triplicate, so they actually look really, really nice in terms of repeatability. And we could quantitate about down to about 1% with this carboxypeptidase digestion. And without it, because it's so complex, we got down to about 5% of relative abundance that was down to reliable quantitation. Okay, so that was intact. Let's move on to a subunit digestion, where we break apart the antibody into basically three 25 kilodalton pieces using an enzyme called Fabricator. It'll cleave just under the hinge. So what we do with that is we do the Fabricator digestion and then do denaturation reduction, or actually partial reduction in this case, where we break them into kind of three identical pieces, two of each of the ones if you're talking about the whole map. So with that, we can do the same sort of quantitation. This is showing you the FC subunit of the N-glycoform distribution, and as well as the C-terminal lysine variant. And I've split them into, on the left side in that pinkish orange color is the ones without C-terminal lysine, and the ones with C-terminal lysine are on the right side in blue. And you can already notice differences there, as well as the different N-glyco profile for each one. You notice that we start to see some of the kind of immunogenic glycoforms for the Remicade and Inflectra, the orange and blue bars there. And that's because they were made in a different cell line than the Avsola and Renflexis. And again, this was about a triplicate injection, so the repeatability is great. And with this one, we can get down to about 0.5% of relative abundance confidently. All right, native ion exchange MS. So this one, I think we hear about a little less often, but it's really useful, is looking at charge variants. And most often you see this not attached to a mass spec detector. Most of the time it's not compatible, the conditions of the separation. So with this, we've developed an MS compatible one using ammonium acetate. For the separation, it's a dual pH and salt gradient. So with this, you see the top right has the overlay of all four of those biosimilars. And those three main peaks really correspond to the C-terminal lysine variants. And then as well as some other things like deamidation and things like that will pop up in charge variant analysis. For more details on this analysis, you can check out the application note there if you're looking to try to set this up. Here we have, like I said, that same overlay. I've just annotated it on the left side. So Remicade in black, Inflectra in green, Avsola in blue, and Renflexis in red. And now that we have the MS attached, we can really dig into each of those peaks and look at it qualitatively to see what's under there. I did note that most of the charge variants are due to the C-terminal lysine. So with one of these, I've taken carboxypeptidase B digest again to remove all the unprocessed C-terminal lysine, injected both of those into our ion exchange method. And the overlay here with the blue trace is the treated sample, and the black trace is the untreated sample. So you see that it sort of collapses up into the main peak. And so most of the basic variants are actually due to the C-terminal lysine. All right, here I'm showing you just an example of some of the MS data that you can see from these peaks. I've chosen a couple of acidic peaks, A2 and A1, as well as the main peak, and then one of the basic peaks just after the main. This is from Remicade, and you can see the combined raw spectrum in the middle of the slide here, and then the Maxent1 deconvoluted spectra that we get. And I just want to highlight a few things here in the blue boxes on the A1 peak, that we can start seeing some of those immunogenic glycans forms. The sialic acid type glycans will come out in an acidic peak. And with MS detection, we can actually see those, as well as the basic peak B1. That's the green one on the bottom. We can start to see some of the +1 +1 lysine with those acidic glycoforms. This slide is showing actually the UV quantitation for each of the peaks in intact form. So the bolded ones, I don't need to go through all of them, but you can take a look. Some of them that I've pointed out already. Those main peaks in bold will show you some of the differences between the biosimilars. Those were just the main ones that came out. So the +0 and +1 and +2 C-terminal lysine species. We can also do this native ion exchange method at subunit level as well. So do the same fabricator digestion that we did, and just don't reduce it. So you get, excuse me. We get on the left part of this slide, you see the overlay from the biosimilars. Again, same Remicade, Inflectra, Epsola, and then Renflexis. And the peaks in the front would be the FC peaks. And you can see again the C-terminal lysine variants. And then the FD and light chain peak is the main one at the very end. And you can start to see some of the acidic variant there. And this is the deconvoluted, the combined spectra. Here will show you that it is indeed part of the FD and light chain, that it's a likely deamidated form of that. So this could indeed be used as a possible screening method for a deamidation of the FD and light chain. And again, same thing here as we take the UV area for quantitation. This is just the table showing the differences, so you can quite easily see some of the differences between your biosimilars in charge variant analysis that you might not see in, let's say, reverse phase. Okay, moving on to a peptide mapping analysis. At this point, I would like to quickly introduce Rapizyme Trypsin, our new trypsin product, starting earlier this year. It's autolysis resistant, heat stable, and highly active. And the reason for that, it's homogeneously methylated trypsin. And the overlay on the bottom here is just showing you incubated enzyme by itself. And so the black one is a competitor trypsin that was incubated for, I think it was, it's a just half-hour incubation for both of these. And you can start to see those black peaks are autolysis species from the trypsin. And the Rapizyme is in blue, and all you see in that one basically is the full length trypsin. So it's highly autolysis resistant, can be used in these protocols in high ratios, which is key here in this analysis. Okay, this is showing you just another Rapizyme trypsin an example. So the top trace is kind of a leading standard method of 1 to 20 enzyme protein ratio of Remicade with a leading MS grade trypsin for 3 hours at 37 degrees. And this is pretty typical. That's kind of the gold standard. We took that same leading trypsin and did that at 1 to 5 enzyme protein ratio for 30 minutes, same at 37, and that's the middle red trace. And then we took our Rapizyme trypsin at 1 to 5 enzyme protein ratio for 30 minutes, and that's in blue at the bottom. And you see that a lot of these yellow peaks are unlabeled or unknown peaks. Those are generally autolysis peaks. To kind of quantitate that a little bit, I took the total TIC area and saw what percentage of the TIC area was made up of expected tryptic peaks. Tryptic infliximab peaks. And what we come up with the standard kind of method with the leading trypsin was about 94%. The one with the leading trypsin at 1 to 5 ratio was 88%. So about 12% of all of these things are unknowns or autolysis species. And then when you do the same thing with Rapizyme, you get 96% of the chromatogram is what you hope it would be. And we get 97% sequence coverage in all of these cases, regardless. Okay, there's those extra peaks just highlighted for you. Okay, looking at that a little bit more in graph form, I know that was looking at chromatograms kind of can be a little tough. So on the left side is, again, that TIC area in bar graph form. So the leading trypsin 1 to 20 ratio for 3 hours, kind of that leading standard is on the left. The middle is the 1 to 5 ratio with the same commercial trypsin, and then Rapizyme is on the right side. And taking a look at the expected peaks is the big green bar, basically. That's just shown in another way. We get 96% of expected peaks with Rapizyme trypsin, and then very little of the unwanted stuff. As far as undesired species go, I took that and plotted it out again with bar graphs and kind of showed what are we seeing here in the unwanted stuff. And for missed cleavages and autolysis, we get almost none with the Rapizyme trypsin in this case compared to the competitor one. And then the non-specific cleavage and the other just unknown or unlabeled things were pretty much on par with the competitor one. Back to our case study with infliximab biosimilars. The top trace of this peptide map is Remicade injection, triplicate injection. So that's really showing the reproducibility of the the LCMS for chromatography. And then the bottom trace overlay is the 4 biosimilars overlaid with each other. You can start to see a couple of little differences here and there, and we'll use Unify to kind of figure out what those are. Okay, this is just showing you the high sequence coverage obtained with the Xevo G3 in MSE mode. For anyone who doesn't know, that's kind of a data independent acquisition. And then we have our Remicade and then our 3 biosimilars on the right side. And if you notice, you take a look at those the unhighlighted species, those are, small peptides that might get lost in the void volume of the chromatography. They're possibly just missed by our own confidence criteria of fragment ions matched. They just may not have enough to generate in the first place. So 97% sequence coverage is pretty good compared to or as expected. All right, this one is a quick example of a oxidized peptide looking at the fragmentation data or high energy data. This one is peptide heavy chain 11, trypsin peptide 11. And you can start to see the different fragmentation pattern, and you can see that it is actually at Y5 where we start to see the oxidation, where that +16 difference is starting to occur in the profile. So you can confidently say it is indeed that methionine that's getting oxidized. Okay. One thing I wanted to point out about the Zevo G3, I think I mentioned it earlier, is it has the Stepwave XS ion guide in it, and that is great for lowering the in-source fragmentation that we see, especially with these labile peptides like glycopeptides. It's pretty common to see on an earlier generation QTOF about 7 to 10% of these sort of oxonium ions where you're losing the sugar from the glycan. And looking at the same thing on the Zevo G3, we get only about 2%, which is great. You're keeping your peptide intact, and you're able to, look at it in a more real way. Okay. One thing also I'd want to point out about peptide analysis, switching gears a little bit to the Peptide MAM, Peptide MAM app. And this is a guided workflow where when Peptide MAM, we're looking at a reference injection as well as, many, let's say, stress studies, or you can use it in our case with our biosimilars, anything that has the same sequence and you want to start noticing differences. So this app will help you to set up your processing methods. You can do acquire and process through it. It sets up system suitability tests and make sure that, everything in your whole queue looks good as far as your suitability goes. You pick your peptide attribute list. So you pick a small subset of peptides that you want to monitor throughout the entire sample set. You pick out your processing criteria, and then you also have the option to do new peak detection as well, which I'll get into at the end. And then, of course, you have your data reviewing and reporting. It'll tell you if your system suitability passes. You have interactive peak review, as well as your results. They have a summary table, and you have the option to export a report as well. For our biosimilars, this is a quick example. What I'm showing here is 4 different peptides or 4 different things that we were monitoring for. I picked the C-terminal lysine, the 2 oxidations, and 1 deamidation. And what you see here is triplicate injections of each one. So between number 1, 2, and 3, that's all Remicade. 4, 5, and 6 is Inflectra. 7, 8, and 9 is Avsola. And 10, 11, and 12 are Renflexis. Those are just the injections. So those should remain pretty much the same across the board, which we expect. And that's good. And then we can take a look at some of the differences. One of them, again, is the C-terminal lysine. Remicade, of course, has the most, followed by Inflectra, then Avsola, and then Renflexis has the least. And that we've already correlated in both our intact analysis and our charge variant analysis. We can also look at the 2 heavy chain peptides that have oxidation on them, both of which are pretty low, between 1 to 2%, but you can start to pick out and tease out minor differences between your biosimilars. And then on the bottom left of the slide is a deamidation on the heavy chain as well. And yeah, Remicade, Inflectra, and Avsola are pretty close. Renflexis, for whatever reason, was a little bit higher. Okay, you can also look to peptide mapping for glycosylation profile. Here, I'm not going to go too much into detail, but we've kind of split them up into the major glycoforms. You can pick out differences and correlate to the intact as well. We have the immunogenic glycoforms and the lower level glycoforms that you can look at peptide level. With this assay, usually about 0.1% relative abundance is what we're looking at for reliable quantitation. Okay, A quick thing about new peak detection. Most of the time, we're going to be doing this in an MAM form, where let's say you do a stress study and you start looking for things that pop up after stress that were not in the reference. For this one, I've also looked at it as far as biosimilars go, because that sequence is going to be the same. So I wanted to see what was different between all of them. So with this, we have a total of 9 new peaks detected with our software. We have no new peaks in the Remicade replicates, which is expected. And the same new peaks were detected in each of the respective biosimilar replicates, which again, very good, as we expect. And so when we do this, we can use our Unify app and our Peptide MAM app to go back and forth to assign new peaks and confirm or reject the new peaks within Peptide MAM. Which is exactly what I did. So here's the plot of what those new peaks might be. The graph here shows the Inflectra had 2 new peaks, and that came down to a possible clipping species and glycation. The Avsola had 5 new peaks, and 2 of those I still didn't know what they were. 3 of them were possible clipping. And Renflexus had 3 new peaks, 1 of each of oxidation, a possible clip species, and an unknown. The bottom of the slide shows how it looks like in Peptide MAM when you're doing the review. You get the extracted ion chromatogram for the new peak, as well as the MS, the scan for it. And then on the right side is one of those, the same peak in Unify, where I went in and confirmed that it was a possible clip species. So in summary today, I hope that I have shown you that the Xevo G3 QTOF in Waters Connect informatics platform, and enables this robust characterization of biosimilars and just biopharmaceuticals in general, I suppose, in a variety of analytical assays. We have the performance and data quality, the automated analytical workflows, and Waters Connect informatics platform for compliance ready. And then we can do with our innovator and biosimilars, the results that we got. The biggest differences were in the N-glycosylation and the C-terminal lysine. Here's the whole list of people that I have to thank for putting effort into this work. Bunch of people here in Milford, as well as a cross collaboration with our UK colleagues as well, and our Milford demo lab as well. I'm not going to spend a lot of time on these. These are just extra bits of information for you if you need. There's part numbers for Rapizyme Trypsin, as well as the columns that were used in this webinar, and a couple of app notes for all of the work that was done here. So thank you all for your kind attention, and I will give it back to Scott.
Thank you very much, Sam. Before we go into the questions that you've asked, my colleagues have asked me to pose a question to you about your interest in what you've heard today and whether you wish to be contacted about any of the products and workflows that we've discussed today. So please, we'll give you the next 20 seconds or so to answer that question as we compile the questions that you have asked to pose to Sam for all the great work that she's shown us over the past half hour. Okay, I see the question has closed. I would remind everyone that this is a biosimilar seminar series, where this is episode 2 of that series. We have episode 3, which really discusses the development of methods during early development that you would use for characterization and maturing those methods into routine assays for late development, manufacturing, and QC. So that'll be episode 3 on October 5th. And then one of our customers engaged in biosimilar production we'll talk about the workflows that they've developed to evaluate their biosimilar candidates during bioprocessing and through further development of those biosimilars. So that's episodes 3 and 4. Again, these will all be recorded. The QR code on the right of the screen can be used on your phone to register for these additional episodes, but we will also have a follow-up email that will have the link for registration as well. Okay, let's see. We have a number of questions that were asked during the session. And let's start with, you had said when you discussed the Xevo G3 system that you can calibrate pretty high up to 16,000 M/Z. How high in mass to charge do you actually see monoclonal antibodies with the native methods you're using, like SECMS or the ion exchange MS that you showed?
Yep, I didn't show a lot of where that's actually being useful, but for this analysis, we look at a monomer of a mAb would be around 5,000 to 6,000 M/Z range, so still within the normal calibration of 8,000. But it's really when you start looking at SECMS for aggregate analysis, where you start to see the dimers and trimers, where they would be up in that between 8,000 and 16,000 range.
Okay, there were a couple questions on digestion. The first of which was about the Rapozyme amounts that you used. What was the amount of mass, I guess, of the target innovator or biosimilar that you used in your digestions that you showed in your data?
Sure. So the digestion itself, I believe we started with somewhere between 50 and 100 micrograms before desalting. So this was a denatured, reduced, alkylated, and desalted sample. So somewhere between 50 and 100 of desalted sample, and then after desalting, check the concentration. And I would take about 50 micrograms out of my that to actually do the digestion with. So for 50 micrograms, 1 to 5, you'd need 10 micrograms of the trypsin. And then as far as the injection goes, it was about, I believe, 200 nanogram on the G3, but I will double check that and get back. Don't quote me on it.
Also in the realm of digestions, so someone's indicating that they do shorter digestions, like 5 minutes, for some quick screening, as well as overnight digestions. Are there people doing this? Are there protocols for doing this with your new trypsin enzyme?
Yep, definitely. We have an app note out that came around the time of launch in March. Shows a couple of different ways that you can do this. With, I think the fastest one that we have in that app note is about 30 minutes with the 1 to 5 ratio digestion. And then all the way up to an overnight digest, where I would take some considerations if you want to go overnight. We lowered the pH a little bit to 6.5 instead of 7.5. So we reduce any of the artificial deamidation that you get with those extended time digestions. And with the overnight digestion, it is a ratio of, I think, 1 to 100 of the enzyme to protein. Yeah, we can,
so can they
send that
actually take their existing methods and just substitute the Rapozyme as a starting point?
Yep, absolutely. It's basically a standard trypsin, but it's not, it's autolysis resistant, which means it also might be a little bit more active, especially if you have a longer digestion. Over time, the trypsin kind of loses activity if it's chewing itself up. So the longer you leave it, if you put it in your same protocols, you might see it working more than what you used to. So if you start to see non-specific digestion, all you need to do is just reduce the time or temperature.
Okay.
Or ratio. There's so many things you can do.
Good. Some questions, a question about reporting. So in your MAM experiments, you showed deamidation as one of the modifications you were monitoring. Do you typically report deamidation as the unmodified percentage and the individual aspartic acid and isoaspartic acid isomeric forms, the two forms you see from deamidation, or do you combine the two deamidated forms when you report deamidation?
So good question. In peptide MAM, you have the choice to do either one. From a scientific standpoint, I would say keep them separate just so that you can monitor them both independently. And in order to do this, I would say you need good chromatography to be able to separate them and be able to confidently be able to monitor and quantitate. So our premier columns for peptide mapping have really helped with that as well. So especially acidic peptides like that have possibly some tailing or whatever, but they they've really helped with keeping those species separate so that you can look at them over a number of samples.
Okay. Another question about MAM. Some of the peptides are reported as potentially clipped peptides, meaning processed, where part of the traditional cryptic peptide would be missing. So how do you distinguish a truly clipped peptide in the sample versus over-digestion artifact or an in-source fragment?
That's a good one too. An in-source fragment would have the same retention time as your precursor. So those are pretty easily noted. It'll also have the same kind of profile for fragmentation. Then if you have, thinking between the digestion artifact non-specific cleavage or real clipping species, to test that out, you could do a number of time points of extended digestion. And if you see that that non-specific cleavage increase over time, then you can say, yeah, it's probably due to my digestion rather than a real clip. But that's how you would go about figuring it out.
Okay. A couple quick product related questions I can probably handle quickly. Sure. Question was, does the Xevo G3 QTOF have ion mobility? And the answer is no. It is a standard QTOF format. The ion mobility systems that we have are the Synapt HDMS system, as well as the cyclic ion mobility system. That one done. And is it possible to fragment a subunit with the Xevo G3 sort of top-down experiments? And the answer is that's possible to do using CID collisional fragmentation. There are some tools within the waters_connect Unify app for looking at top-down type work. But some of the other platforms we have, such as the Synapt and the Cyclic, have alternative fragmentation, electron-based fragmentation, that also is pretty useful for doing those top-down experiments. So one more for Sam. And if there are any other questions, please feel free to open up the Q&A button at the bottom of the screen and pose them. This one's a pretty in-depth one. So someone interested in quantification of their variants. And they're trying to put together the values you gave. And you said, from the intact spectra, you can see down to about a 5% level of a variant. at about 0.5% for subunits. And how does this compare to the peptide MAM technique you talked about? And if you're talking about things like glycopeptides or glyco variation, released glycan workflows.
Yep. So like Scott said, and like I mentioned in the presentation, at intact level, I would say reliably down to 5%. You can start detecting things under that, but as far as, you know, repeated injections and things, you might get a little bit more variation. And then same thing for subunit at 0.5% relative abundance. But then when you get down to peptide and release glycan, those are typically in the 0.1% relative abundance area. So, you know, the smaller you make it, the easier it is to quantitate lower.
Yep. Of course, the complexity of the experiment increases, is the cost for digging deeper into those lowest level variants. And the final question that I see we have on our board today was about the MAM workflow, and how reasonable is it to use MAM to assess host cell protein levels?
Oof, that is a question that's not for me, unfortunately. We might have to follow up with you afterwards.
Okay, I can probably answer. I have attended the MAM consortium meetings, as well as worked on the people who have developed our MAM tools. And I think the consensus is moving away from MAM as a tool to look at host cell proteins, at least in the polished product, where they're at typically single digit or lowest, 10 ppm and below. It probably doesn't have the ability to validate host cell proteins at those levels that you're seeing them in modern products. But when it's used in development and process development, when those host cell proteins are at higher levels, it may prove a useful workflow, I think, is the consensus that we're seeing build within a lot of the speakers at the MAM consortium and scientific meetings. Okay, are there any other questions? I do not see any other questions on the board. So I want to thank Sam for her presentation and for answering the questions that you posed. Remind everyone, again, this is a series with 2 additional episodes coming over the next couple of weeks, but this is a recorded series. You can review any of the episodes that are produced, as well as refer your colleagues to them if you think they'd find them of interest. And there will be a link in the follow-up email for you to register if you haven't made use of the QR code that's to the right of this slide. So saying that, thank you all for your attention and joining us today. And we look forward to seeing you at the future episodes of this series. Thank you.